start because I think Amina is it's bedtime for her. So it's good that she's made it. Let the others join as and when. I know it's a little disturbing, but can't help it. So we have today's slide, which is titled, this topic is the body's warriors. And I think you all know how important these cells are to us. So in this presentation, you will review the 5 different types of WBCs. Who is it now? Let's start. All right, 5 different types of WBCs, which you all know: neutrophil, basophil, eosinophil, lymphocyte, and monocyte. And the development, that's leukocytosis. Maybe it's a little revision for you. Maybe this leukopoiesis is a little bit of a boring thing, but you need to know, especially if you're going to work in the hemat lab, especially when you're going to deal with certain types of leukemias. Function of each cell is also described in short in this presentation. Phagocytosis, one of the most important functions of a neutrophil, is vividly explained. And I think you like what you see there. So we come to the function of each cell. You've got the neutrophil. Main function is phagocytosis. If you're making notes, put it down. Eosinophil is anti-allergy. It's a granulocyte, and you can see the granules. You've got basophil, again, the function is anti-allergic. Then you've got the lymphocyte. It's an agranulocyte. Take a look at it. And the main function is the production of antibodies, the T cells and the B cells. And you've got the monocytes. The function is their macrophages. So they get rid of certain entities which your body and mind does not like. And what the neutrophil cannot do, the macrophage does. So we ask the question, what is leukopoiesis? So leukopoiesis is a form of hematopoiesis in which the white blood cells are formed in the bone marrow, located in the bones in adults, and the hematopoietic organs in the fetus. And I think you know, blood formation takes place in the fetus right from the first trimester. Then it becomes organoid, your spleen and all that takes over, and then the bones, the long bones and the flat bones. White blood cells, indeed all blood cells, are formed from the different and pluripotent hematopoietic stem cells, which give rise to several cell lines with unlimited differentiation potential, right? Because this goes on in your body from the day you are born to the day you die. These immediate cell lines or colonies are progenitors of red cells, erythrocytes, platelets, megakaryocytes, and 2 main groups of blood cells, the myelocytes and the lymphocytes. On the basis of history of association, leukemic diseases is divided into 2 main groups, acute and chronic. We'll talk of that in another lecture. The incidence of both chronic and acute leukemia is higher in males than in females. Some say it has to do with a little bit of hormones. Let's see, many causes of leukemias. So let's start with the first maturation, the myeloblast. So the blast, as you know, is the most immature of cells. And blasts are a little difficult for you and me to say, oh, this is a neutrophilic blast, or this is an eosinophilic blast. Guys, you can hear me properly? Any problems with the audio?
No, there is no problem.
Okie dokie.
Everything is okay.
That's
All
fine.
right. Okay. So we'll
Now,
continue.
okay, can hear. Yeah.
All right. So I especially if you are my student, you will remember how boring this was. So the cell is about 10 to 12 microns in diameter. Nucleus is round and stains purplish blue. Definite nuclear membrane is present. And usually we have a saying that says a blast is identified by the company it keeps. So around the blast, you have neutrophils, so you presume this belongs to the neutrophil series. Or you find lymphocytes, so you can presume that by this is a lymphoblast. Chromatin strands are delicate, well defined, and evenly stained. Two or more nucleoli can be seen. Moderate amount of non-granular cytoplasm, which forms a thin rim around the nucleus, is present. So you can take a look at the three cells which are in front of you. You've got the myeloblast, and you've got the promyelocyte and the myelocyte. So, what are you doing? All right, so the second stage is promyelocyte. The cell is 15 to 18 microns in diameter. Nucleus is round or oval and stains purple. Nucleoli, if present, are not well defined, and the chromatin is coarse. There are varying numbers and sizes of dark, non-specific granules, which stain from red to purple. They're azurophilic, bright blue in color, like a cloudy sky. Cytoplasm is blue, lighter staining adjacent to the nucleus. You can take a look at the three stages up here on the right-hand side. The third stage is the myelocyte. The nucleus is round, oval, or flattened. On one side, stains dark purple. Chromatin is coarse. Early myelocytes may have visible nucleoli, and you can see it in this cell that's in front of you. The cytoplasm is pinkish blue. The specific granules become definite and so numerous that they obscure nuclear details. This is a stage where the differentiation takes place. Let's go to the fourth stage. So now you have the neutrophilic myelocyte. So this, you can guess that this is going to be in the neutrophilic stage, and you can see some neutrophils around it. The first site of neutrophilic myelocyte differentiation or dawn of neutrophilia takes place in the form of a small, relatively light island of ill-defined dark pinkish granules, which develop with bluish cytoplasm among the dark specific metachromatic cells. A characteristic change in the color of staining carried out in biological tissues, exhibited by certain dyes when they bind to particular substances present in these tissues called chromatophores. As the myelocyte ages, the dark granules become less prominent and the specific neutrophilic granules start becoming predominant. Nucleus is round and the nucleoli may be indistinct or absent, and chromatin strands are thick. You can see the cell on the right-hand side. Stage 5 is neutrophilic beta-myelocyte. Now, you have to be an expert to see this and guess what it is. The nucleus is dark purple, slightly indented, and has a small pinkish blue granules. They are slightly smaller than myelocytes and have smaller nucleus and less well-defined chromatin structure. Cytoplasm is pink blue, and they are found in contents of myelocytic hyperplasia. And you can see the cells here. You've got the specific granules, you've got the neutrophilic metamyelocyte, and you've got a couple of them around. And take a look at the size. It's still on a larger side. Okay. And then you've got the band, the neutrophilic band, which you and I do get to see in a normal person's smear. About 4% is acceptable. Nuclear indentation becomes slightly more marked. You can see it in this picture. More than half of the width of the round nucleus. Nucleus is dark purple, band-shaped, and chromatin is coarse and stains blue-black. There is usually dark cyanotic mass At each pole of the nucleus, cytoplasm is blue-black. So what is this ionotic? Is the irreversible condensation of chromatin in the nucleus of a cell undergoing necrosis or apoptosis. And this cell you will find in the smear. And what is the significance? In case of infection, where the WBC count is high, the neutrophilic count is high, you may get more than 4% bands. So when you say 7% bands or 8% bands, 10% bands, then the doctor knows that there is some sort of pathology or infection or inflammation in the individual. And this could be anything. And then you come to the neutrophilic segment cells. You can see them here. The nucleus has 2 to 5 lobes connected by a very narrow filament. Nuclear indentation is greater than 1/2 of its diameter. You can see the cells here. Stains dark purple, chromatin is heavily clumped, and the nucleoli are absent. Cytoplasm stains light pink or to bluish and has many small evenly distributed pink to rose violet granules. They are twice the size of red cells. So you can take a look. Those of you who have left your practice, take a look because it's very easy to diagnose. What happened? Who's just close to us? Yeah, take a look. All right, back to the. All right, so we have the 7th stage. We've taken a look at the 7th stage. We'll come back to it. So this is done. Now we come to the lymphocyte. The lymphocyte and the monocyte have 3 stages of development. I haven't gone into eosinophilic and basophilic development. This is more or less the same. And of course, the granules give away whether this is a eosinophil or a basophil. So we come to the lymphocyte, 3 stages. You've got the lymphocytes, the prolymphocytes, and the mature lymphocyte. So here we have lymphoblast, which is preceded by a lymphoid stem cell. Cell size is 10 to 20 microns. Cytoplasm blue, scanty, no granules or rods are never present. Nucleus, purple smooth chromatin, round central or eccentric, 1 to 2 nuclei. The nuclei to cytoplasm ratio is 4:1. And you can take a look at the cell in the center and at the bottom. And it's the lymphoblast, it will tell you that there is something abnormal in the cell. Maturation sequence continues, pro-lymphocyte, difficult to distinguish, as you can see. Size is 9 to 18 microns. Blue scanty, cytoplasm is blue scanty. Usually granules are absent, but a few azurophilic granules may be present, and we are seeing this more and more these days. Nucleus, purple condensed chromatin, round or indented eccentric, 0 to 1 nucleoli may be present. The ratio is 4:1. And then you have the mature lymphocyte, which I'm sure all of you have seen. All lymphocytes are derived from the lymphoid lineage, constitute 20 to 30% of blood leukocytes, continuously circulate in the blood, and are capable of migration into tissue spaces and lymphoid organs. Small lymphocytes have high nuclear to cytoplasm ratio and lack cytoplasm granules. So you have the B lymphocyte and the T lymphocyte, B coming from bursa, T from thymus. Large lymphocytes can be twice the size of small lymphocytes and contain granules. You can see it on the right-hand side. These are also called large granular lymphocytes, LGLs, and include activated T cells and plasma cells. Plasma cells are activated B cells, and the natural killer cells are NK cells. And this is a very beautiful description of the monocyte. Again, three stages: monoblast, promonocyte, and monocytes. So the monoblast, round to oval, may be irregularly shaped. Nucleoli 1 to 2 may be visible. Chromatin is fine, as you can see. Cytoplasm blue to gray. Granules none. In the promonocytic stage, you've got the nucleus, which is irregularly shaped, folded, may have brain-like convolutions, and this is very, very characteristic even in the mature cell. Nucleoli may or may not be visible. Chromatin fine to lacy. Cytoplasm light blue to gray. Granules free azurophilic. Vacuoles may be present. The mature monocyte variable may be round, horseshoe shaped, often has folds producing brain-like convolutions, and this is quite diagnostic. Nucleoli not visible. Cytoplasm lacy. Cytoplasm is blue-gray, may have pseudopods. Granules, many fine granules, giving the appearance of ground glass. And you can especially see it in this right-hand slide that you have with you. Vacuoles absent to numerous. Take a look at that presentation on the right-hand side. It gives you a very, very good picture of the nucleus, the cytoplasm, the vacuoles, and the ratios. Okay. So then we come to the lymphocyte. Agranulocytes, which include lymphocytes and monocytes, are a type of white blood cells that, unlike granulocytes, lack visible granules. And you can see that. As such, they have a clear cytoplasm that allows for better visibility of the nucleus. Apart from lacking clear visible granules, agranulocytes also characterized by a single large nucleus. The nucleus is not lobed. And guys, here I want to tell you, I'm just refreshing you. Usually the size of the red cell is comparable to your small lymphocyte, about 6 to 8 microns. This is how we take a look at the smear and say, do you have microcytes, very small cells? Or you have macrocytes like you do in some anemias, large cells. So, usually microcytes are with iron deficiency or TALs, and your macrocytes are with your vitamin B12 type of deficiency. So keep this in mind. If you're going back to the lab, compare the size of your red cell to the lymphocytes. I'm talking about small lymphocyte, okay? And coming back to monocytes, for this reason, they are also referred as mononuclear leukocytes, which differentiate them into multi-lobed granulocytes. In the body, agranulocytes are involved in adaptive immunity, acquired immunity, and thus use immunological memory to act against invading pathogens. So right now, I won't go into the types of immunity, which they're like. And that especially when we talk of delayed hypersensitivity, this comes into play. Maturation, back to it, during the production of monocytes, the hemocytoblasts first differentiate to produce myeloid stem cells. The myeloid stem cells differentiate to form monoblasts, which is committed to developing into a monocyte. Here, the monoblast further develops into a promonocyte, which produce the monocyte. Hence, they leave the circulation and enter into tissues. This is one of their properties. Monocytes become macrophages that are described as big eaters, especially if you have infections like parasitic infections. You will find that the monocyte count increases, especially when you have things like malaria and so on. Okay. We'll continue. In the production of lymphocytes, the hemocytoblast first differentiates to produce the lymphoid stem cell. In turn, lymphoid stem cells differentiate to produce the lymphoblasts. So, series of proliferation and differentiation, the lymphoblast produces pro-lymphocyte, which in turn develops to produce a lymphocyte. There are two lymphocyte types of lymphocytes, the B and the T, and B comes from bursa, T from the thymus. Being the mature lymphocyte, the B lymphocyte leaves the bone marrow and enters the bloodstream through the sinusoidal capillaries before reaching the lymphoid organs. The T lymphocyte, on the other hand, is not functional and has to reach a lymphatic gland, thymus, in order to mature. And here we have a very sweet picture telling you about the functions of the granulocyte. You've got this guy who is fighting infection. Some of the main functions of a granulocyte include protect body against invading microbes, bacteria and viruses, destroying cancerous cells, activating the other immune cells, destroying old and damaged cells. And we come to phagocytosis. Pay attention to this because this is one of the most important functions of the neutrophil. Phagocytosis is a process by which a cell uses its plasma membrane to engulf a large particle more than 0.5 microns, giving rise to an internal compartment called the phagosome, which is a vacuole in the cytoplasm of a cell containing a phagocytized particle enclosed within a part of cell membrane, and you can see it in the pictures. It is one type of endocytosis, the taking in of the matter by a living cell by invagination of its membrane to form a vacuole. Some of the cells that use phagocytosis include macrophages, protozoa, and so on. So take a look at endocytosis. On the right hand, there's a little table there. You've got pinocytosis, plasma membranes fold inwards, forming a pinocyte vesicle. Then you've got phagocytosis, plasma membrane engulfs the solid material, like you see in the cell on top. Receptor-mediated endocytosis, specific molecules bind to specific specialized receptors. So this is a very interesting slide. So keep it in mind or make a note. You have my PPT with you. So here you've got phagocytosis. You've got this guy opening his mouth, wanting to swallow that little bit of trouble which is in front of him. Okay. Now take a key look at these 6 stages. So what happens? Phagocytosis. The leukocyte absorbs bacteria. Can you see the bacteria going into the neutrophil? Leukocyte ingests bacteria. It's being eaten up. Leukocyte expands from ingesting large numbers of bacteria. And lysis takes place. You can take a look at the cell, the 4th cell below. White blood cells lyse, releasing cytokines, chemical signals, which cause local inflammatory reactions. It's a cascading effect. You can't remove this. This little part down here. All right, and then you've got the 6th, you've got cytokines attract new leukocytes to fight bacteria. So you've got the pus formation that may happen in certain infections, and you can see that the neutrophils are torn apart. You're going to repeat the cycle, okay? Come on, next one. Again, now this is nicely described here. Activation. This step of phagocytosis occurs when the cell comes in close proximity to given objects or particles. See the first one. For instance, when phagocytes are near such cells as bacteria, phagocytes, this activates the phagocyte and stimulates binding, which is happening in the 2nd cell. The first step also involves chemotaxis. Okay, so you've got the formation of the phagolysosomes and the destruction of the viruses taking place in the cell. The binding takes place. Here, surface receptors of the phagocyte bind, adhere to the surface of the particle. This step of phagocytosis is necessary for the molecule to be ingested. Keep this in mind. Receptor binding is an important step in phagocytosis, allowing the cell to identify the object, particle, bacteria, etc., and thus elicit the appropriate reaction. So, here, what is this guy telling you? I'm not just any phagocyte. I am a professional phagocyte. So, this is the job. Again, that picture repeats in front of you the 6 stages. Then, in the third step, ingestion takes place. The cell phagocyte starts expanding as it surrounds the molecule. The process also involves formation of a vacuole or vesicle around the molecule as it is completely ingested, and you can see it in the pictures. In the first picture, you can see this antibody-dependent cellular phagocytosis, ADCP as we call it, and you can see the antibodies around the target cell. Then you have the formation of the lysosome, the middle, can you see it? And then you have the lyse target cell, and then this is thrown out and the cell destroys itself. Phagolysosome. Keep that in mind. The fourth step is digestion and destruction. In some cells, enzymes in the vesicles, like lysosome, break down the molecule into simpler components. Waste materials that cannot be used are then removed from the cell through a process known as exocytosis. However, the phagocytes involved in immunity, special structures are known as peroxisomes. Peroxisomes are oxidative organelles, are created to trap and remove the toxic molecules. So you've got the endocytosis, then you've got the exocytosis. So what is ingested inside has to be thrown out. And this is what happens here. You've got the peroxisome, you've got the lysosome, and which is made up of cholesterol, and eventually what is ingested is thrown out. That's the last slide. You would like to see some pictures again? Hello? So what I've given to you today is in a very, very simple form. We'll take it further in the next lecture coming up next Thursday. There is a